Forensic Science Lecture 9-4, Forensic Characteristics of Semen. In dealing with sexual offenses, there are many cases that are actually received by forensic labs involve sexual offenses, and it makes it necessary to examine different items for seminal stains. And that's a two-step process. First, you got to locate the stain, and then that stain needs to be subjected to tests that will prove the identity of the individual who deposited them. Often seminal stains are visible. They exhibit a stiff, crusty appearance to them. However, if the item has been washed or only contains a minute amount, there's little chance for visual detection. And so the best way to locate and characterize a seminal stain is to perform an acid phosphatase color test. And so this test involves acid phosphatase, which is an enzyme that's secreted by the prostate gland. The presence is detected when it comes in contact with sodium alpha nephyliphosphate and Fas blur B dye. And what happens is it turns purple. So the sodium alpha nephyl phosphate is a acid solution, and so when combined with the Fas blur B dye, it emits a purple color to it. Also can be detected when it comes in contact with 4-methyl umbilical phosphate, or MUP, and it will fluoresce under UV light. Fluoresce meaning emit a light or a glow. Reaction time of less than 30 seconds is a strong indication that semen is actually present. Another test for examining semen is through a microscope. It can be identified by the presence of spermatozoa, which are sperm. They're slender, elongated structures. They're about 50 to 70 microns long, each with a head and a thin flagella, or a tail. The sperm is actually located by immersing the stained material in water, stirring it rapidly, and placing a drop under a slide or on a slide, and then dyeing or staining it. And then it can be examined under a microscope. Chances of finding the sperm should be good, since a normal male will release anywhere from 250 to 600 million sperm per ejaculation. But it's not always true. Sometimes sperm binds very tightly to cloth. It can also be extremely brittle when it's dry, and it could disintegrate. It can also disintegrate if it's washed or rubs against other objects. And crimes may involve male with low sperm count, which is a condition called oligospermia, or it can involve a man with no sperm at all, which is called aspermia. Aspermatic male are actually increasing in number due to the popularity of vasectomies that are occurring. So just because there is no semen, or excuse me, there is no sperm present, doesn't necessarily mean that there hasn't been a sexual crime that occurred. There is another test which is called the prostate specific antigen, or PSA, and when acid phosphatase is detected but no sperm are located, forensic analysis need to perform this other test. And what they're looking for is the presence of a protein that's called P30, or prostate specific antigen, and it's unique to seminal plasma. A positive P30 test with an acid phosphatate color test reaction of less than 30 seconds indicates unequivocal identification of the semen. Once semen is identified, the next task is to associate with an individual. The test for P30 uses an anti-P30 that's produced using the rabbits in the same way that the drug antibodies and the human antibodies were formed for testing of blood type or the presence of drugs, which we've discussed before. And it uses electrophoresis machine where they place the anti-P30 in one well of the plate and the extracted stain in the opposite. And when electricity is applied, if semen is present, the antigens and antibodies will move towards each other and form a line. Seminal constituents on a rape victim are very important evidence to obtain, and it's not always present. Physical injuries such as bruising, bleeding can also confirm a violent assault. Forceful physical contact between a victim and an assailant may result in the transfer of physical evidence. And we know this, physical evidence such as blood, hair, fibers, we know this as Lockhardt's exchange principle. And so because of that, to protect all evidence, possible evidence, all outer clothing and underwear must be carefully removed and packaged separately in paper bags, again, not plastic, because it is biological and has the potential of growing bacteria and so on. Along with the outer clothing and underwear, also the bed sheets can also be packaged and brought for analysis as well. Collection of the rape evidence involves the rape victim undergoing a medical examination as soon as possible to collect key evidence. The evidence that's collected from the rape victim include pubic combings, a pubic hair reference sample, external dry skin area swabs, vaginal swabs, cervix swabs, rectal swabs, oral swabs, and swabs of body areas such as the breast that the assailant may have bit or licked, or anything that may have transferred DNA to them, along with head hair, blood samples, fingernail scrapings, and of course all clothing, and a urine sample. Time is key in a sexual assault case due to the fact that the seminal acid phosphatase in the vaginal cavity decreases with time, and there's little chance of identifying any seminal acid phosphatase after 48 hours after intercourse occurred. P30 is normally not detected in a vaginal cavity beyond 72 hours. And so it's key to quickly undergo the medical examination. Once a suspect is apprehended, hopefully within 24 hours of the assault, it may be possible to detect the victim's DNA on the suspect, either in the underwear or on the penis itself through a penile swab. Other evidence that's collected from the suspects include, again, all of their clothing or his clothing, pubic hair combings, head and pubic hair reference samples, and then blood sample or a buccal swab, the swab from the inside of the cheek. And hopefully they'll be able to link the evidence found on the victim with the suspect, and if not, then that may exonerate that individual. This is the end of 9-4 lecture. There will be a quiz next class.